Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear students, we are studying the chapter that is Principles of Inheritance and Variation. Today, we are starting with the post Mendelism. Post Mendelism means the era after Mendel. Means actually we call post Mendelism as it starts after the rediscovery of Mendel's work in 1900 onwards. Means after the rediscovery of Mendel's work, which is done by three scientists in 1900, that is Carl Collins, Igor Deriz, and the von Teschemark. After the discovery of that, the era of post-Mendelism started. Now in post-Mendelism, we are going to study two things: one of the allelic gene interaction and one of the non-allelic gene interaction. But in today's lecture, we are mainly focusing on the allelic gene interaction. Non-allelic interaction we will study in the later part. Okay, so let's understand what is allelic gene interaction. Allelic gene interaction means here it is the alleles of one gene, how the alleles of one gene get interacted, that we are going to study. So this allelic gene interaction is also known as one gene interaction, or we can define it as a interaction between alleles of one gene, and how they interact with each other, that we are going to understand. In this, we are first was one more thing I want to mark you here. What things we are going to study in this particular part? So in this, we are going to study different things, and in different things, we are going to study incomplete dominance. Okay. Second, we are going to study codominance. Third, we are going to study multiple allele. Fourth, we will study lethal gene. And fifth, we are going to study pleiotropy. Okay. So this many things we are going to study in this lecture. Okay. So first we are starting with the incomplete dominance. This incomplete dominance is also named as intermediate inheritance, and it is the first exception of the principle of law of dominance. It is the first exception to law of dominance of Mendel. Because here law of dominance expresses complete dominance of one allele over the other. But here the complete dominance is not observed, incomplete dominance is observed, so it is an exception. Now, how this is an exception, that we are going to study. So it is obvious deviation from Mendelian concept of dominance. Okay, and if we talk here, then this incomplete dominance, when experiment on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. Means the F1 progeny is not similar to the parent. Why? Because in complete dominance we have studied that if we make a cross of tall and dwarf in F1, you will get tall only. which means it is resembling with the one of the parent. But in incomplete dominance, it is not going to resemble any of the parent. But the trait which will be expressed, that is of another form, which is an intermediate one between the two. So means in these, the dominant allele of a character is not fully dominant over the recessive allele. So in heterozygous condition, an intermediate mixed form will appear. What will happen? Intermediate mixed form will appear. So if we talk example, so good example to understand incomplete dominance is color of the flower in dog flower called snapdragon. So if we talk about snapdragon or dog flower and its color, the question is what is the scientific name of this snapdragon? Then it is Antirrhinum majus. And if we talk about a dog flower, then in this snapdragon, there are a flower color is taken as a character. But this flower color, if you take, if you take a flower color, then three colors are seen. One is red, one is white. These two are parent, and intermediate which is obtained, that is pink. So pink is the intermediate form, or the form or a trait which is of mixed type or in between the traits of parent. Okay, so that is the one example. So if we talk about antirrhinum species, in this you can see this is antirrhinum flower. You can see one is a red flower, one is a white flower, and you can see the pink flower are obtained. So this shows the intermediate or mixed type characteristics. Okay, so this is nothing but what parents. These parents are red color, so we represented genotype as capital R, capital R. These are or white color, so we represent as small r. And this is of F1 progeny. This F1 progeny is of particular pink color, and the genotype is capital R, small r. So this is showing the intermediate or mixed type. This means this is showing the incomplete dominance. Okay. Now, if you make a cross of capital R with the small r, red flower, white flower, and we get a capital R, small r, that is F1, that is the pink colored, we have discussed just now. When the F1 are self-pollinated, the F2 resulted in the following ratio. If you carry out a cross of F1 selfing, then you will get a capital R, small r in 1 is to 2 is to 1 ratio. And red, pink, and white is also in 1 is to 2 is to 1 ratio. Means here the phenotypic ratio, if we carry out a selfing, then we, for F2 generation, then we, what do we do? Capital R small r crossing with the capital R small r. Then progenies will be capital R is to capital R small r is to small r small r. So here genotypic ratio is 1 is to 2 is to 1, and phenotypic ratio is also 1 is to 2 is to 1. Okay, so that is the thing. Now after this ratio, next we talk about this is the particular showing a same thing in the snapdragon, or we can say. Now see, if we talk about some another example, and if we talk about the discovery, then this is also showing a, you can say, the particular red, white, and pink. So in this cross, genotypic ratio were exactly as we would expect in any Mendelian monohybrid cross, but the phenotypic ratio had changed from 3 is to 1 to 1 is to 2 is to 1 ratio. What happened was that R was not completely dominant over small r, and this made it possible to distinguish capital R small r as a pink from the capital R red and small r white. Thus, the phenotypic and genotypic ratio in F2 progeny are the same, that is 1 is to 2 is to 1. Okay, so this is the thing. Now, The other characters in which the incomplete dominance is known to occur, that other examples are which show the incomplete dominance, that is flower color in Mirabilis jalapa. Mirabilis jalapa is a 4 o'clock plant. Okay, and this has a three tint: red, pink, and white. Pink is of intermediate form. Okay, pink is of intermediate form. Okay, now if we talk about some of the MCQs, what is the scientific name of cerebral? Yes, before that, one more example I am referring here. Just you remember these two that are enough. The first, the flower color in Snapdragon. Another example is undulation falls. That is a feather color. Undulation falls. That is the particular type of, we can say, a duck species. Near three traits are obtained: black, gray, and white. Okay. Okay. So this is the thing which occur in the in this main characters in complete dominance is C. Now, after this incomplete dominance, if we take some of the questions, what is the scientific name of Snapdragon? Then we have discussed it is antirrhinum major, so it is antirrhinum species. Okay, first is the correct answer. After this, what is the phenotypic ratio in incomplete dominance? In incomplete dominance, phenotypic ratio is 1 is to 2 is to 1, and genotypic ratio is also the same one. Okay, and then we are going to discuss a codominance. Okay, if we talk about codominance, so co-dominance is another exception to the law of law of dominance. Here also, complete dominance does not occur. Okay. In previous incomplete dominance, what capital R, small r? Small r is obviously not expressing, but capital R was not completely able to dominate over the small r. Because of this, it is not showing a complete rate. It is showing a slow version, that is a pink, and that is formed due to the mixing of red and pink. Here, the both genes show equal expression. Okay, here both genes show equal expression. So in codominance, if we say, then if we define it, then in this, both the alleles of a character are equally dominant, are equally dominant. So in heterozygote, heterozygous condition, both will express both will express simultaneously or side by side. Okay, so in this, both the alleles of a character are equally dominant. Okay, but there is no mixing of character. So if they are equally dominant or equally expressing, means no allele is going to be dominant over the another allele. Means there is neither dominance nor recessiveness, only equal expression of both the alleles will be seen. So this is the codominance. In case of codominance, the F1 generation resembles both the parent, not anyone, both the parent. And in incomplete dominance, it is not resembling anyone. Okay, example of the codominance are ABO blood grouping in human being. Okay, ABO blood grouping in human being is one of the important example. Okay, we will discuss that. Another is the seed coat pattern and size in lentil plant. If we talk about some more examples, then we can write coat color in kettles. That is also one example. Okay. MN blood group. One more type of blood group is there. That is known as MN blood group, which is a rare one. And sickle cell anemia. That is also showing a codominance. Okay, so we will discuss that. So first we are taking a coat color in kettles. You can see one coat color, one is black and one is white. You can say black or brown, we can say. And in the third one, you will see the white coat also as well as black. See, if you can see here, it is a coat color that is which one? White. If I give a genotype white as a B1 B1, and if this one is considered as brown or black, some kettles have a black hairs over their coat, some have white hairs over their coat. If the coat is white, then the white coat, this is a brown or black coat, we can be there. I am giving it as a B2 B2. Now in this, they have a hairs of white color as well as a blue, black or brown colored hairs are there. So this is the F1, and this F1 is showing the black and brown both hairs for the body. So it is known as roan, which having a black and brown both hair or this one. If you do selfing of this, if we perform selfing of this F1, so F1 is a roan. So here genotype will be B1 and B2. So if selfing of B1 B2 is done, then B1 B2 will cross with B1 B2. So here the gametes will be B1 B2. Here also B1 B2. If a cross is made, this is going to be B1 B1. This is going to be B1 B2. This is going to be B1 B2. And this is going to be B2 B2. Okay, so this is going to be what? White. This is what? Brown or black. And these two are of roan. So genotypic ratio is 1 is to 2 is to 1, and phenotypic ratio is also 1 is to 2 is to 1. Okay, so that is the thing. In cattle and horses, if you cross a pure red and a pure white, you will get a RW which produces a color road, same color which is observed in cattle also. Okay, so that is showing a codominance means equal expression both the genes. Okay, now after coat color, if we take some more examples, then we have discussed that AB blood group is there. In AB blood group, ABO blood grouping actually AB blood group in human being is there, and for AB blood group, IA and IB alleles are there, and this both produces A and B antigen over the RBC. Because of this, they determine the AB blood group. So means both are expressing equally, so it is a codominance. Okay. Another example we have discussed MN blood group. In MN blood group in humans, which is a rare, in this you will see that there are two alleles LM and Ln. Okay, so if LM allele is in homozygous condition, then it is a M type of blood group. If Ln in homozygous condition, that is then it is N type, and if it is a LMLN, then it is Mn type. Okay, so this three type of blood group are there, but in this one show the equal expression or codominance. Okay, apart from that, another example is sickle cell anemia. In sickle cell anemia, you will see that there are two genes, HbA and HbS, two alleles. HbA, hemoglobin A, is normal and HbS is defected. So the defected hemoglobin will not able to carry the oxygen properly, and the sickle shaped RBC will be obtained. So here also this both will express in heterozygous condition. Okay, means they show the what particularly codominance, and we will discuss the sickle cell anemia in the later part. Okay, so this is the difference between codominance and incomplete dominance. Okay, now we are going to talk about some of the questions, and here one question we are going to deal is that what will be the F2 phenotypic ratio, what will be the F2 phenotypic ratio of a dihybrid of a dihybrid cross in which one character show codominance and and other character show incomplete dominance. Show incomplete dominance. Okay. So if you want to answer it, what will be the F2 phenotypic ratio of a dihybrid cross in which one character show the codominance and another show the incomplete dominance. Okay, we have to identify phenotypic ratio. So in codominance, the phenotypic ratio is 1 is to 2 is to 1, and in incomplete dominance, which is occupied or which is shown by the another character, the phenotypic ratio of this is also 1 is to 2 is to 1. So if we make a cross of this, the phenotypic ratio in F2 generation, which will be obtained here, that is going to be 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1, and that is going to be the answer for this question. Okay, now next. Okay, so this is the okay, dear students. So this is the thing about the codominance. Now next thing which we are going to understand, that is a multiple allele. Now see, what do you mean by multiple allele? Means the gene is only one, but in spite of having a two allele, they have many. So multiple allele is defined as presence of more than two allele, presence of more than two allele of a character is known as multiple allele. Means they have more than two forms. Suppose we have studied that particular capital T and small t, they are the two alleles only. But by chance, if they develop as a capital T, small t dash, capital T dash, so they have four alleles. Okay. So alleles are obviously going to be present at the same locus on the homologous chromosome. Okay. But here more than two alleles are present for a one gene, for coding for the one character. If we talk about this in more detail, then we come to know that all these alleles arise by mutation. All these alleles arise by mutation. Mutation is responsible for this. Here, this all alleles are present at same locus on homologous chromosome. Okay. Now it is known that in any diploid organism, in a diploid organism, only two allele of a character, only two allele of a character can be present. And rest of the other alleles and the rest of the other allele can be present in a population. Can be present in a population. Thus, multiple allele can be studied by studying population genetics. Okay. So multiple allele can be observed or seen in when a particular we are considering a population. In one individual, we can't see a multiple allele of one gene. Okay, so that is the one of the important, you know, thing. Okay, now if you talk about examples of this multiple allele, then we come to know that the first example for this is ABO blood grouping in human. ABO blood grouping in human have three alleles for a gene I. Okay, that is IA, IB, and I naught or I0. We will discuss this in detail. Second example is self-incompatibility in tobacco. Self-incompatibility in tobacco. And it is controlled by 4 to 6 alleles. Third character is coat color in rabbit. In coat color in rabbit, there are 4 alleles. And fourth one is eye color in Drosophila. Eye color in Drosophila is controlled by 15 allele. Okay, so these are the four examples which show the multiple alleles mechanism, in which we will discuss ABO blood grouping first. Okay, every example is not important to consider, but first two we will discuss in detail. So first we are going to talk about ABO blood grouping. Now, dear students, this ABO blood grouping in human, you know that it is discovered by Landsteiner. And for identification of ABO blood grouping, there is a presence of specific antigen on RBC surface. So these groups are assigned based on presence of specific antigen on RBC surface. And this antigen are produced by I gene. I stands for isoagglutinogen. Now this I gene, if we see, which is present on chromosome number 9, this I gene have three alleles, and this gene is present on chromosome number 9. The three alleles which are present for the I gene are IA, IB, okay, and I naught or I0. IA is a dominant allele which code for A antigen. It forms A antigen. This is going to produce B antigen, and this do not form any antigen. Okay, so this is the three alleles which are present for the I gene, which determine the blood group. Now, if we talk further, then we come to know that the blood groups which are produced here, that are of four type, that is A, B, AB, and O. Now in this blood group, if we talk, the possible genotype of their alleles in diploid individual can be, if diploid individual have a IA, then they can produce A in homozygous condition. If there is IA and I naught, then it show a dominance, complete dominance, and show a blood group. Same way, genotypes for this are IB, IB, and IB, I naught. Here genotypes are IA, IB, and here I naught, I naught. If we talk about antigen which are produced here, the antigen which are produced here are of A type in A blood group on the RBC, B type on the B blood group, and both A and B type on RBC, then that determine the B blood group, and we have studied that this show codominance as well. And here no antigen. But against the antibody which are present in that blood, that are B antibody, A antibody, then no antibody, any antibody is not present against the AB antigen. And if any antigen is present, they react with this one, and the agglutination, blood clotting will occur. And here both A and B antibody are present. So this is the thing. Now, in this genotype, if we see, total six genotype are possible because three alleles are there. One, two, three, four, five, six. Six alleles are there. Okay, and six genotypes are produced by the three alleles. So six genotype are possible. So if somebody directly asks that the particular trait or particular character or particular gene having this many alleles, then how many genotypes are possible? Such type of question some can ask. Then what you will do? So there is one formula that will help to understand or calculate this type of questions. So what question can be there? Question is, if particular character is controlled by a gene having 10 alleles, how many genotypes are possible? If this type of question you will get, for this you need to remember one formula, and that formula is total possible genotype is equal to n into n plus 1 by 2, where n is equal to number of allele. So if we solve this question, 10 alleles are there. So if you apply this, then 10 into 10 plus 1 by 2. So it is going to 10 into 11 by 2. So it is 110 by 2. So total 55 genotypes are possible. Okay, so such type of question you can answer by like this. Now, we are in a continuation of AB blood group, and you know that the A blood group, B blood group is there, AB is there, and O is there. In that we call AB as a universal recipient, and we called O as a universal donor. Okay, I'm replacing it here. This O is universal donor. So A is recipient, so it can receive from A, it can receive from O, and it can receive a blood from B. But O is donor, so it can give A and B as well, and they all can give themselves similar type of blood group. Okay, so this is the thing. Now, one more factor is that is known as RH factor. This RH factor is present on RBC surface, and this RH factor is there, and its antigen on RBC surface, RH antigen, which is coded by this gene, is present. This RH antigen is also known as D antigen. If RH antigen is present, then it is positive. If the antigen is not found, then it is negative. So if RH antigen is present on RBC, then it is positive blood group. If RH antigen is absent, then it is going to be negative blood group. Now it is known that if we talk about positive and negative, then positive can then give themselves, negative can give themselves, but negative can give particularly positive, but positive cannot give a negative. Okay, so this is how the blood transfusion can. Okay, after this we are going to solve some of the questions, and in that if we take a question, the question is which of the following parental combination will will never produce a child of O blood group. The options are one, A and B parent. Second option is A and O parent. Third option is AB and O. Fourth option is B and O. Now in first option that can be IA I naught and IB I naught. So if I naught will merge, then O blood group is possible. If it is IA I naught and it is I naught, then also O is possible. But if it is AB, then there should be a IA and IB reacting with I naught. Then only possible combination is IA I naught or IB I naught. Means A and B blood group are possible, O is not possible. So that is going to be the answer. But we check here as well. If it is IB I naught and I naught, then yes, O blood group is possible. Okay, so B, C is going to be the correct answer. The third is going to be correct answer. Okay, dear students. So this is the thing. Now after this, in ABO blood grouping, we are going to discuss another example, that is self incompatibility in tobacco. Dear students, in tobacco, if we talk about this self incompatibility, in this, what happens if pollen grains are particularly of same genotype are able to germinate on the stigma of the same flower and able to develop a pollen tube, then it is known as self incompatibility. The rejection of self pollen from germinating on stigma of the same flower is known as self incompatibility. Now it is regulated or controlled by S gene, and S gene have a 4 to 6 alleles like S1, S2, S3, S4. So if suppose this is a stigma and this is the flower, you can say, okay. So dear students, this is regulated by these four alleles. Suppose this is a flower, okay, and this flower has a genotype of S1, S2. Then this is also going to produce a what? The pollen grain of S1 and S2 type. So this S1 and S2 type of pollen grain, if fertilized here, they are not going to develop this particular pollen tube. But if another flower is there which has a genotype S3, S4, S3, S4, and it produces a S3 and S4 type of pollen grain, they are not going to pollinate this self, but they are able to pollinate this flower with the S1, S2 genotype. So S3 or S4 can pollinate this. So the self pollen is not going to what a compatible or going to develop a pollen tube. So this rejection is controlled by we can say the multiple allele phenotype, and you can see there are so many alleles are there. Okay, dear students. So I hope you understood this in a very well manner. So in this lecture, we have covered with the initial part of incomplete dominance, then codominance, and then multiple allele. Now in allelic gene interaction, two things are remaining, that is the lethal gene and the pleiotropy, that we will cover in the upcoming lecture. Okay, thank you.